The Mediterranean, the meeting place of civilizations, the scene where Western culture arose and grew, and where drama and tragedy also write their pages. A place where myths and history intermingled to form a common past and present. It remains, nevertheless, a great unknown when we immerse ourselves in it. Three continents embrace the Mare Nostrum, but its history and legends have told us little of what lies beneath the surface. When we dive, we witness a grandiose spectacle of shapes and colors through which nature expresses itself in virtuous harmony. A very rich biodiversity. It recreates a universe of species of breathtaking beauty. Among this enormous and surprising explosion of life, we observe close to the coast a mollusk, standing upright on the seabed, recognizable in its shape and, at the same time, largely unknown. This bivalve, the largest in the Mediterranean and one of the largest in the world, can measure more than 1 meter and live for more than 40 years. Its name is fan mussel or Pinna nobilis. The survival seemed assured by the fact that they had few predators and were not part of the food and economic chain of the human species. But it is not always direct human action that is the main threat to biodiversity. Nature tends to self-regulate and establish its own mechanisms of balance and survival. But this does not mean that it is safe from processes that become serious threats. Pandemics with very serious consequences also arise in the natural environment. In 2016, a parasite spread across the Mediterranean basin with such enormous speed and aggressiveness that 99.9% of the fan mussels specimens were wiped out in a short period of time. Millions of specimens died with no time or opportunity to prevent what was in fact presented to us as a case of extinction of an animal species. It is at this point that science comes into play. The IMEDMAR of the Catholic University of Valencia, whose marine station is located in the port of Calpe, brings together its scientists and coordinates an international LIFE project to try to reverse this phenomenon.
Science refuses to stand idly by and watch the disappearance of species. Specifically, in the case of the fan mussel, Pinna nobilis, the fact that we have found some specimens resistant to the plague is very important. And this is indeed the starting point of our research. Knowing the causes of this resistance, the fan mussel habitat, its physiology, its genetics, its biology, and any other aspect that help us is crucial in order to safeguard our seas from the disappearance of this emblematic species. Moreover, scientists and science know no borders, and we always work together to try to tackle these complex problems of species loss, as they are transnational. International research projects are necessary to tackle the problem of species loss. The Pinnarca project is an international project whose objective is to try to prevent the extinction of the fan mussel, the bivalve mollusk, the largest in the Mediterranean, which, due to a parasitic disease that began in 2016, has lost practically all of its populations in the Mediterranean Sea. It is a project whose objectives also include trying to disseminate and make the species better known to the general public, and also to carry out an exhaustive study of the populations and individuals that are still alive in order to work with them and try to recover and reduce the rate of disappearance of the species. It is being developed with the contribution of the LIFE program, which is the European Union's financial instrument that supports the development of environmental and nature conservation projects and climate actions. As we have said, it is an international project in which countries such as Spain, Greece, Italy and France are taking part. In Spain, the coordinators are the IMEDMAR Institute of the Catholic University of Valencia and also the University of Alicante, the IRTA Institute of San Carlos de la Rápita, the CSIC through the IEO and the IMEDEA, the NGO Ecologistas in Actions in Murcia, and then in Italy, the University of Studies of Naples Frederick II, in France, the Paul Ricard Oceanographic Institute, and in Greece, the University of the Aegean.
On the Mediterranean coast of France, a team of specialists from the IOPR is working on one aspect of the research, genetic research.
In the European Pinnarca project, here at Paul Ricard, we coordinate all the genetic studies, but we do also other things, larval collectors and also population sampling. As part of the LIFE Pinnarca program, the Paul Ricard Oceanographic Institution and the Toulouse Science Faculty, with the IMO laboratory of Stéphane Coupet, my colleagues are in charge of the genetic part of this program. We have sampled individuals in particular. We have taken tissue samples from resistant or non-resistant individuals in populations found in particular coastal lagoons, and we also have samples from the open sea. The aim of this genetic part is first of all to define a little bit the genetic structuring of the residual populations, and also to define if we can actually find an introgression of Pinna rudis in the resistant Pinna nobilis. So there we have the analysis of the first 6 samples that we have. Can you explain to me what we see on the screen? You had given me 3 individuals potentially contaminated by the parasite, 3 healthy individuals, and the molecular analysis shows that the 3 dying and sick individuals are indeed contaminated by the parasite's DNA. There we can clearly see the increasing curve, which reflects the specificity of the parasite's DNA in our sample, whereas the healthy individuals are completely negative for the parasite. Correct? So there we have the T1, T2, T3, and then the negatives that are not yet out of the 44 cycles.
Erta La Rapita, the Institute of Agri-Food Research and Technology, is located in the east of Spain. They carry out studies on the surviving fan mussels of the Ebro Delta, one of the only two populations surviving in sanctuaries in Spain, with the intention of safeguarding this important core population of the species. To this end, they collaborate in the environmental improvement of the delta by installing traps to retain excess sediment, study the environmental conditions of the delta, and carry out direct actions to collect larvae by means of collectors and translocate individuals from vulnerable areas to safer ones within the delta itself. They also participate in the global studies on the pathology that has affected fan mussels and wiped out their populations. Removing fan mussels from their natural environment to study them in the laboratory is not an aggressive action and is the only way to learn about the parasite's action and chances of survival. We moved to the eastern Mediterranean, to Greece, on the island of Lesbos, where the University of the Aegean is located. The study of living colonies and survival conditions, as well as the movement of larvae, are the objectives of their study.
Okay, in the Marine Biodiversity Lab in the University of the Aegean, we study for many years the populations of fan mussels in Kaloni Gulf and in Yera Gulf. These two gulfs are in Lesbos Island in the northern Aegean Sea, and they used to have very large populations of fan mussels. From one of these gulfs, the Yera Gulf, the population has become extinct, totally extinct, but there are still live individuals, many live individuals in Kaloni Gulf. So in the framework of the Live Pinnarca project, we try to rescue this population. So we conduct census to estimate the magnitude of the population. We also put larval collectors trying to study the recruitment of the species, and if we manage to collect larvae, to try to make them survive, and after reaching a specific size, to transplant them. And we also try to, together with the marine authorities and the management body of this Natura 2000 area, we are trying to impose additional restrictions for some activities that are distractful for the fan mussels. During the Live Pinnarca project, we conducted many censuses all over Greece, trying to investigate if live populations of fan mussels exist. And Orestes, together with Vangelis, conducted all these surveys, and Orestes could tell us about what they found.
Yes. In total, more than 160 surveys were conducted across the Greek coastline, both in the Aegean and the Ionian Seas. Alive Pinna nobilis were found only in the semi-enclosed gulfs of Kaloni, in Lesbos Island, the Aegean Sea, and in Ambrachikos Gulf in Ionian Sea. The characteristics of the gulfs maybe could limit the Aplosporidia infection to Pinna nobilis. In Ambrachikos Gulf, around 50% mortality was estimated. All the specimens recorded were estimated to be young adults. And in Kalonic Gulf, we had around 90 to 95% mortality. More than 70% of the live individuals were estimated to be recruited in 2022.
In Marmara Sea, due to low salinity and the specific conditions that are prevalent there, fan mussels are protected from infection from this parasite. So we are interested to investigate if from the outflow of the Marmara water to the North Aegean Sea, also Pinna larvae arrive in the North Aegean and settle. For this, we have developed, our physical oceanographers have developed oceanographic models to investigate the fate of the water that flows from the Marmara Sea to the North Aegean. And we have put collectors, larval collectors, in sites of high probability of arriving larvae, trying to investigate if this hypothesis of the Marmara Sea acting as a donor site for the North Aegean is actually true.
In eastern Spain, the University of Alicante, with the participation of the Aquarium of the University of Murcia, is working on improving the maintenance of fan mussels in captivity. The work also includes the study of the Mar Menor lagoon, the second of the last two fan mussel sanctuaries in Spain, where it is essential to monitor the environmental parameters and control the survival conditions of the fan mussels, which are strongly affected by the pressure that humans exert on the lagoon.
Well, of all the ones we have here, I think this is the oldest one, which is from 2019, May 2019. Do you remember where it was taken from? This one, yes, from El Barón. Yes, that is correct. El Barón. Actually, I remember that I have all the location data, the natural environment, everything. One interesting thing is also the epibionts that have grown, because with the previous study that we had, the Dicedia was different from the Mediterranean Dicedia. So you also have it as a reservoir of, yes, apart from that, they work very well of invertebrate species in the lagoon, and the filter feeders are there. That's great. And this one is a bit more modern. This one is from 2021. It came from the ones we took from El Pedrucho during the anoxia process. Yes, and well, this one, as you can see, also has a polychaetes. It also has hydroids in there, which have to be cleaned from time to time, because they can block. Well, this is from the Pedrucho, which we took out in 2021, in November, but it was just after the summer. There was anoxia there, and even hypoxia. Yes, even hypoxia in that survey. I was there too. You were there too, and we were there, so this is the one left from that time. This one has recovered well. The others that came here in ruins, the poor things. This is the one that deceives, that is buried deep in the sand and looks like it's smaller. And when you pull it out, we saw that it was, well, almost 70 centimeters. Yes, exactly. And it's one of those that have been used to check the healthiness of the fan mussel. Yes, it was with Patricia and Francesca. We took out the hemolymph, and it seemed that everything was fine.
In the Balearic Islands, the IEO and the IMEDMAR, which belong to the CSIC together with the LIMIA, use larval collectors and research the important field of reproduction. The key is not only to keep alive the maximum number, of specimens, but also to recover the populations from natural reproduction.
The area we wanted to discuss with you are about the resistant ones which are in the Cabrera National Park, the capabilities of hosting other species. Then it is a zone of humidity that is a little below sea level. There are salinity points up to the connection with the sea. From this northern part of the lagoon there is a gradient. But it does not have several points, here, and this area here, and they reach up to 2.5 meters deep. In other words, there are several marine species. Yes, there are marine species. I think it could be a very interesting place to put seeds, and of course, perfect for growing them. From the Balearic Islands Oceanographic Center, we are involved in the LIFE project as a partner of the CSIC together with IMEDMAR. From here, we're leading this partner, and specifically, we are coordinating a total of 3 actions. IMEDMAR coordinates the action of the larval collectors, and we coordinate A2 actions, which is the search for optimal sites and the monitoring of the same. And also D2 actions, which is the follow-up of resistant individual, that is to say, their follow-up in the open sea, in the Mediterranean, and the resistant individuals in the sanctuary area.
Laboratory work with Pinna is complemented by field work. Observing breeding populations and collecting larvae is an essential task.
Hola, Amalia.
Hello, Amalia. How are you? Hi, how are you, Maite? Have you been able to look at the last samples I sent you? Yes, in fact, I am looking at them right now, and I attribute this to the diet, which is not ideal.
Hola, Gaetano.
Hi, Gaetano. Hi, hello. How are you? Listen, I'm bringing you the biopsies of the other resistors that I told you we had to take samples from. Okay? So, I'll leave them with you, and they're all yours. How did the analysis of the others go? We did 2 kinds of analysis. One by analyzing the samples, doing mitochondrial DNA analysis, and they came back practically all Pinna nobilis, except for one that came back Pinna rudis. And then we did a nuclear DNA analysis.
In Pinnarca, we are responsible for C1, Action C1, which are the larval collectors. These larval collectors are put in the water in the hope to catch the recruitment of the pen shell, because if there are reproductive adult populations, we will see larval recruitment, which we hope to catch in the collectors. So it's important that there are very many collectors out there in the Mediterranean, so we have a higher probability of actually catching these recruitment events. If there are adult populations that are reproducing, the larvae will be in the water column, and having more collectors in the water column enhances the probability of actually seeing them. So what we do is we send the material of the larval collectors to the other partners within the consortium, so we have a homogeneous design throughout the Mediterranean. This assures that we can compare the results between the larval collectors of all the partners. We haven't been able to find a lot of recruitment up till now, which was kind of expected, but unfortunate. But this year in 2023, there actually has been some recruitment, which actually pinpoints at reproductive populations of the adults, which is very hopeful.
We return to IMEDMAR on the western Mediterranean coast to learn about aspects of its daily activity.
Here at the IMEDMAR marine station, we mainly keep groups of Pinna nobilis individuals, both juveniles and adults, to carry out different experiments on them, both for feeding in order to keep them as long as possible, to improve the quality of life, and when they are in tanks, and for reproduction purposes, to try to reproduce the fun mussel or to treat the disease. It is also very important that if we can treat the disease, to try to do so in the specimens that are infected. For example, there are times when, from the collectors that are installed to recover larvae from the sea, the juvenile specimens are infected, and we're trying to treat them with cold water to try to cure them. In addition, we have a series of adult specimens that we keep in tanks with 1,000 liters of water with a closed circuit, where we control the temperature, the concentration of oxygen, the food is given in calculated doses throughout the day, and these specimens are given temperature treatment so that they can mature and reproduce. We also have a small room, some aquariums where we have some juveniles that have been here for almost a year and have now grown to almost 12 centimeters. In order to be able to feed all the specimens, we have now some tubes where we have phytoplankton. There are 40-liter tubes, about 20 tubes in total, and so we can feed them every day as we produce this phytoplankton. On the other hand, we also work in the field in various parts of the project, such as surface censuses up to 20 meters, deep centers up to 40 to 45 meters to see if there are any survivors of Pinna nobilis, to locate optimal areas to reintroduce new specimens of Pinna nobilis, and we also place collectors to catch larvae and work on the translocation of individuals. That is to say, moving individuals from one place to another to improve their survival. And finally, we're making a great effort in dissemination to show the world the problem, the importance of the species and all the work that is being carried out with the Life Pinnarca project.
The disappearance of a species is a disruption of the natural balance and is a serious matter that affects us all. Science puts knowledge, personnel, and facilities in place to prevent such dramas.
Having achieved parasite-resistant specimens opens a great door of hope for scientists. Since we are able to reproduce these resistant specimens in captivity, we open up the possibility of developing new specimens and being able to reintroduce them into the natural environment.
Nothing in nature is alien to us, and everything in nature interacts. We know from science that human activity is altering the fragile natural balance, and that pollution, the excessive use of natural resources, and climate change are altering the conditions of habitability of the planet, deteriorating the survival capacity of humans and many animals and plants on which we depend. Only the decisive action of scientific institutions and citizen commitment will be able to stop a process that could become irreversible and disastrous for our own species. Institutions provide resources and facilitate the coordinated action of the scientific community. Citizens inform themselves, promote dissemination actions, and support the action of science and institutions.